Oh, yeah.
Hit.
Wait, what?
At the end of the hour, the last thing I said was, I couldn't think of the word I was looking for. I said, when you guys play tug of war, and I couldn't think of spirit games. You have all played tug of war before, correct? Or witnessed someone play tug of war? And when both teams are pulling equally, that flag that's at the center of the rope is aligned in the middle, right? Yeah. That's how the game works. And it stays aligned perfectly in the middle so long as both teams pull with the same force. That's what happens here. The centrioles who have migrated to opposite sides of the cell are pulling with the same amount of force on those chromosomes. When they pull with the same amount of force, because they're attached on two different sides, the chromosomes align at the metaphase plate. The only time they're going to move from there is if our chromosomes separate into sister chromatids. Then, when that force is applied by the centriole and it pulls, then the sister chromatids migrate to opposite sides of the cell. That's when we've moved on to the next phase. So here, we're looking at metaphase. What's the next phase? And we still have all the same parties, all the same players. We still have a centriole on either side. We still have spindle fibers. Except now, we no longer have a centromere. That's because the centromere has dissolved. The centromere dissolves, which allows those the centrioles who are pulling so very hard to pull sister chromatids to opposite sides. God bless you. There's still no nuclear envelope. There's still no nucleolus. Still no nuclear envelope, still no nucleolus. They're not completely migrated yet. So this is like the process of moving from one to the other. This is the in-between stage. At this point in time, the cell looks like it has twice as much DNA as it needs. And it looks like that because the DNA is no longer stuck together in the middle. It has enough DNA for two separate cells. It's the last phase of mitosis. And what should telophase look like?
Two separate cells in one.
This is what's kind of weird. Most pictures are going to show the sides of the cell squeezing in at this point. That's not technically part of telophase. It occurs simultaneous or at the same time as telophase. So it happens at the same time, but it's not a sign of telophase. What is our chromosomes have completely migrated to opposite sides of the cell. They're done moving. So they finish their migration, and what reestablishes itself? A nuclear envelope. So we're getting a new nuclear envelope. This happens at the same time as this, but is different than this particular thing. Let's make it blue so you can see it. The edge of the cell membrane is going to start to indent. And it indents further and further and further until those two indentations touch each other. That indentation is called a furrow. And it's kind of a strange word, but if you think of the contexts in which you've heard furrow before, the most likely one that you have encountered it in is if someone has furrowed their brow. What does it look like for someone to furrow their brow? Makes that wrinkle, right? That indent. That a furrow is an indentation. That's what that word means. So in this instance, we call it a furrow because it's an indentation in the plasma membrane. That process that occurs simultaneously to telophase is called cytokinesis. It's when the cytoplasm is divided in half. So mitosis is dividing a nucleus in half to make 2 new nuclei. Cytokinesis is dividing all of the rest of it in half. So all of the cytoplasm. That's when mitochondria and ER and everything else that you find outside of the nucleus gets distributed to 2 new cells. We call those 2 new cells daughter cells. Even though the parent cell doesn't remain, we still call it 2 daughter cells. So 1 cellular division results in 2 daughter cells.
Does cytokinesis take place during prophase?
It takes place during telophase.
Or sorry, yeah.
Yep. So it kind of sort of starts during anaphase and then is definitely noticeable during telophase. You will not, however, see a furrow in a plant cell. Furrows are unique to animal cells. Why can't a plant cell make a furrow?
Because of the cell wall.
The cell wall. The cell wall is too rigid to allow the plasma membrane to pinch in. So instead, plant cells will do something like this. Here's my square plant cell. Here's the 2 new nuclei. A plate appears down the middle of them that we call the cell plate. And the cell plate is what divides a plant cell in half. So an animal cell gets a furrow, a plant cell gets a cell plate. Everything else will be the same except that last step. And it's important because their cell plate also has to include a new cell wall. So that's also being established there. Every phase but mitosis is called what? Interphase. Has how many parts?
3.
What are those parts? Once interphase is over, we enter what? Mitosis. Is there a particular way we can abbreviate mitosis? Because I'm lazy. Capital M. Sometimes called M phase, which to me, might as well just write mitosis if you're going to write M phase because it's the same number of letters, but who am I? Mitosis gets divided into what parts? And it all ends with the division of the contents of the cytoplasm, which we call Is that as bad as we thought it was? Pretty easy, right? It's just a matter of getting some familiarity with some vocab words. That should, and the reasons why cells divide. So the process of cell division and the reasons that cells divide. Let's see if I can't find you.